Hello and welcome to another episode of Research Talk. Today we are delighted to have Miss Keerthana T V, who hails from Kannur, Kerala. She completed her BS dual degree at IISER Pune, and she is currently a research scholar at the Indian Institute of Science, Bangalore. Her research focuses on cancer immunotherapy using synthetic antigens with a strong emphasis on enhancing immunoresponses through smart molecular design. In today's talk, she will present her work, which entitled as Enhancing TN Antigen Immunogenicity via Albumin Hitchhiking. Notably, her work received wide public attention and has been featured in the leading newspapers such as the Times of India, The Hindu, and edX, highlighting both its scientific as well as societal impact. So, Ms. Kirtana, welcome to the talk, and the screen is yours.
Thank you, Mahesh. Thank you for your wonderful introduction. Hi, everyone. I'm Keerthana. So, today I'll be presenting my work, which is enhancing TN antigen immunogenicity via albumin hitchhiking. So, cancer is one of the devastating health disease faced by the humanity for the past few decades. And according to statistics by Globocan 2021, it is expected that by 2050 there will be almost 35.3 million new cancer cases and almost an 18.5 million new cancer deaths. Hence, there is a growing demand for cancer treatment options. The traditional cancer treatments are surgery, chemotherapy, and radiation therapy, but it has its limitations due to relapse, toxicity, and drug resistance. Hence, an alternative therapy is immunotherapy, which uses the body's own immune system to fight cancer. It works by stimulating the body's system's ability to recognize and attack cancer cells by drugs or vaccines. The main bottleneck in immunotherapy is to figure out the specific cancer biomarker. So, tumor-associated carbohydrate antigens or TACAs are one such biomarkers. As we all know, the cells are covered with a thick layer of glycans, which is called glycocalyx, which is mainly composed of glycolipids and glycoproteins. And these carbohydrates in normal cells are long and branched, but in cancer cells, these are short and unbranched. It arises due to the mutations in the genes, which causes these truncated, heavily glycosylated cancer specific patterns, which will act as an early biomarker and also as a promising candidates for carbohydrate based cancer vaccine design. So here I'll be mainly focusing on TN antigen. So TN antigen is also a tumor-associated carbohydrate antigens, which are truncated or glycans, like found in cancer cell. It is highly expressed in breast, colorectal, and pancreatic tumors, and it arises due to the mutations in the genes for T synthase or cosmic. So chemically, a TN antigen is galactosamine molecule which is glycosylated onto serine or threonine moiety of mu-C1 glycoprotein. So since this TN antigen has its being a promising candidate, it has its own challenges due to its low immunogenicity and immunotolerance. Hence, approaches were carried out to increase this immunogenicity of TN antigens. So, as we all know, lymph nodes are the critical sites for immune cell activation, as most of the immune cell resides within the lymph nodes. So, one way to increase the immune response of these antigens is to directly deliver these to the lymph nodes. And one strategy of doing so is by using a strategy called albumin hitchhiking. Albumin hitchhiking uses the serum albumin as a carrier to improve the delivery of therapeutic agents to lymph nodes. So, in albumin hitchhiking, once the vaccine is injected into the body, the molecule gets incorporated with the serum albumin non-covalently, and it is the intrinsic property of the serum albumin to carry these molecules to the lymph node for antibody production. So this is a work by DJ Irvin and co-workers, where they have covalently modified the CpG molecule with serum binding molecules like cholesterol, monoacyl lipid, and diacyl lipid. And through IVIS imaging, they have observed that most of the molecules are sequestrated into the lymph node when compared to other molecules. So the diacyl lipid is shown to be an effective lymph node traces when compared to cholesterol and monoacyl lipid. Hence, we have chosen our molecule as DSP PEG 2000. So DSP-PEG 2000 is a phospholipid which is known to bind to serum albumin. The serum albumin has 7 fatty acid binding sites. Hence, the albumin binding domain, which contains this ISI lipid, can get incorporated into the serum albumin and can be carried to the lymph node efficiently for albumin hitchhiking method. So, this the diacyl lipid gets incorporated into the serum albumin, and while the PEG block remains exposed, leading to the formation of an aggregate. So this is a brief objective of my work where I'll be synthesizing the TN antigen and TN body PTN antigen glycolipid, followed by biophysical and biochemical studies to study the interaction of TN glycolipid with serum albumin, followed by biological studies in Vivo to study lymph node targeting and antibody response. So these are the molecules which are used in the study, where TN antigen is covalently conjugated to a TSP-PEG 2000 via an amide linker, and to track these molecules to the cell, we have covalently tagged this molecule with a body P tag, forming a body PTN glycolipid. To compare the immune response, we have used a conventional vaccine conjugate where TN antigen is covalently conjugated to BSA. To check the immune response elicited by the linker, we have also used DSP-PEG 2000 alone. So since once these molecules are synthesized and it is in hand, we have moved on to the self-assembly of TN glycolipid. So since these molecules are amphiphilic in nature, it can undergo aggregation in aqueous solution. So we have checked the morphology of these aggregates formed by using AFM and TEM images. From AFM image, we could see that the TN glycolipid forms micelle-like structures, and on introduction of PSA to this particular glycomycel structure, there was a disappearance of these micelle-like structure, which could be a case that PSA has a strong interaction with the glycolipid, hence disrupting this micelle, hence a disappearance of peak. To further study the self-assembly of TN glycolipid, a fluorescent microscopy study was carried out by using DPH as a probe. So DPH in aqueous environment does not exhibit fluorescence, but when it is intercalated into the bilayer of the micelle, it shows fluorescence. From this study, we could observe that the CMC of DSP, the TN glycolipid was 500 nanomolar, and to check the interaction of serum albumin with the glycolipid, we have added serum albumin to the particular solution, and we could see there was no increase in fluorescence up to 1:1 concentration. That is, below 1:1 concentration, there was clearly a disruption of micelle, hence no increase in fluorescence. But once it reaches above 1:1, there was an increase in an increase in fluorescence and formation of micelle. To further confirm this, we have added excess BSA to the same solution, and we can observe a plateauing of peak, from which we can infer that there was a disruption of micelle. So from this, we can say that there is no fluorescent increase or micelle formation when the concentration of VSA is greater than or equal to the concentration of TN glycolipid, which indicate that there is a 1:1 ratio in BSA-TN glycolipid binding. Since it has to be translated in vivo, the abundance of albumin in blood or interstitial space guarantees that no more than one glycolipid will be present on any given albumin. To further study the interaction between the TN glycolipid and BSA, we have done an SPR study using a CM5 chip, which is carboxymethylated dextrin chip coated with BSA covalently. And as a analyte, we have passed TN glycolipid below its critical micelle concentration to remove the complexities due to multivalency. So this is a particular sensorgram where the baseline is due to the binding covalent modification of PSA to the chip. And once we inject the TN glycolipid solution, there is an increase in the response unit, which reaches an equilibrium. And once we stop passing the glycolipid, there is a dissociation phase followed by the regeneration of chip using lysine HCl. And it was observed that the TN, this is a particular sensogram that we have obtained from the SPR studies, and it was observed that the TN glycolipid exhibited relatively a faster association and the slower dissociation rate constant, indicating a stronger binding of lipid chain to the BSA. Since this molecule has to be injected in vivo, the wall of the body or the cells which are lining the body should not be modified with this particular conjugate, since it has a lipid chain and the cells are also covered with a lipid bilayer. Hence, a preferential binding study was conducted in the presence of BSA and cells. So HeLa cells were used, and HeLa cells were treated with body P attached TN glycolipid in the presence and absence of FBS and was monitored over 3 different time intervals. So FBS is also a serum albumin. So when in the presence of FBS, most of the compounds are taken by the FBS, hence unavailable for the cells, hence there is less fluorescence. But when there is no FBS in the media, most of the cells takes up these molecules and shows a fluorescence. And this fluorescence increases with time. From this study, we can infer that the FBS has stronger affinity to the compound when compared to the cells. Now, to check the immune response elicited in vivo, the molecule was injected into C57BL/6 at 3 times at 2 weeks interval, and blood was withdrawn after 1 week of injection. And from these are the 4 groups which are analyzed, where group A was injected with PBS, which will serve as a blank, and group B, which was injected with the DSP PEG 2000, which will serve as a negative control, and group C, which was injected with a conventional TN-BSA conjugate, which will serve as a positive control, and group D, which was injected with the molecule of study, which is TN glycolipid, which will serve as our molecule. And all this positive and the molecule of study was given as a 1:1 emulsion with a CFA adjuvant. And the key players of immune response are antibodies, and these are the main antibodies which are present in the body, and we'll be focusing now mainly on IgM and IgG. So on primary antigen exposure, there is a clear increase in IgM, and the IgG response are delayed. But on secondary antigen exposure, the IgM level remains the same, but the concentration of IgG was raised to a higher concentration, which is called the secondary antibody response. And these kind of immune response are very critical for cancer immunotherapy. From this, so we have already injected the mice with the conjugates, and we have the serum which is isolated from the blood, which has the antibodies. Now, to detect and quantify the antibodies in the serum, we have used an assay called enzyme linked immunosorbent assay, or ELISA, where an ELISA plate, which has 96 well plate, are coated with the antigen, that is the TN antigen, and followed by an incubation with the primary antibody, which is the sera antibody. And to detect this primary antibody, we have used a secondary antibody with an HRP conjugate. If the primary antibody is specific to the TN antigen which is bound onto the plate, this primary antibody will bind to the plate, which is further detected by the secondary antibody with an HRP. After the incubation of HRP, we will add the substrate solution, which is TMP, and the HRP will catalyze the reaction of this substrate to a colorimetric product, which is further detected using a plate reader. So the more the optical density of this product, more will be the binding of this primary antibody. So from the ELISA studies, these are the results that we obtained. So from the graph, it is very much clear that the PBS and the DSP PEG did not induce much immune response, as the optical density is very less. But in the case of TN-BSA and TN-DSP PEG, that is TN glycolipid, the immune response is definitely high. So if you see first and second immunization, the TN glycolipid exhibited a higher immune response when compared to the conventional TN-BSA conjugate. But in the third, after second immunization, there was a clear decrease in the immune response presented by the TN glycolipid, which is due to another phenomena called accelerated blood clearance phenomena. Since a molecule has a PEG linker, which is polyethylene glycol, in the body there will be anti-PEG antibodies which is released, which will eliminate these molecules when injected after third immunization. Hence, there is a decrease in immune response. Now, to further quantify the antibodies in serum isolated from the mice, we have done a serial dilution experiment using ELISA technique. And for this, we have used two serums, which is serum from the mice injected with a conventional TN-BSA conjugate and the serum from the mice which is injected with TN glycolipid. So after the serial dilation, we could observe that the serum from TN-BSA injected mice could detect the TN antigen which is coated onto the plate even after 1:6400 dilution. In case of TN glycolipid, this could detect TN antigen even after diluting it to 1:51,200, which clearly indicates a higher concentration of antibodies present in the serum injected by TN glycolipid. Now to check the albumin hitchhiking property of this molecule, we have done a IVIS imaging where the mice was injected with a body P type glycoconjugate and were sacrificed for organs like heart, spleen, liver, and lymph nodes. And we could see that the most fluorescents were mainly occupied in the lymph nodes when compared to heart and spleen, which clearly indicates an albumin hitchhiking phenomena. To further confirm this, we have done an immunohistochemical staining where the mice was injected with the TN glycolipid and the organs were sacrificed and the tissue sections of each organs were prepared, and if any antigen was present in the particular tissue section, it was stained with a primary antibody, which is a serum antibody, followed by a detection with a secondary antibody having a FITC probe. So FITC is a fluorescent probe, hence more the amount of fluorescence, more will be the amount of TN antigen present in the tissue sections. From the confocal images of the tissue section, we could see that there was an accumulation of molecules in the lymph node when compared to other organs like liver and spleen, which clearly indicates an albumin hitchhiking phenomena. So this is the overall view of action where the molecule which is subcutaneously injected undergoes demycelization in the presence of albumin. It migrates with the albumin which is drained to the lymph node where it activates the immune cells producing antibodies. So in conclusion, the work demonstrates that albumin complexation of TN antigen is an effective approach to site-specific delivery and antibody production. The study establishes TN glycolipid albumin complex through a series of biophysical, biochemical, and biological investigations. The study demonstrates a novel strategy to enhance potency of carbohydrate vaccines, relying on albumin as a carrier to deliver antigen at the target site. Thank you.
With that, we come to the end of today's episode of Research Talk. Thank you, Ms. Keerthana, for such insightful and engaging presentation and for sharing your exciting work on the enhancing cancer immunotherapy through synthetic antigens. If you enjoyed this session, don't forget to like, share, and subscribe. We'll meet you in the next episode, next session. Thanks for watching.